Hello, good morning, good afternoon, good evening, wherever you are. Welcome to another session of DNA rejuvenation technology. And today's session is purification of EVOSK virus by ion exchange chromatography, also called IEX. IEX. Okay, remember in the previous video we purified up to 90 to 95% of purity. In this occasion, we are going to purify from 95% of purity to almost 100% of purity. You see here, in the previous step, we left the AAV-OSK virus almost clean. You see here, there are some contaminants, just a few, and in this step of purification we are going to clean it all. Yay! Oh yeah! Okay, so that is what we are going to see today. We are going to see how to clean it to perfection, to almost perfection, to almost 100%. Ready? Let's go for it. Well, before starting, we are going to gain some context, okay, in case you have just landed here, okay? And I want you to know what it's this all about. Remember the DNA. The DNA is a molecule that is pretty much like this, okay? You know it. You know it. You know it. Okay, the DNA, the storage, the information necessary to form the blocks of your body, pretty much doing everything, okay? So the DNA is kind of the manual, a manual to build things in your body. This is the DNA, you have it here, okay? You see these young people, these smiling, lovely young people? Okay, that is the DNA of these people. You see here, there are little dots, little marks, okay? So this is kind of another layer of information over the DNA. When you have only a few marks, the DNA is young, and as you get older, so here you see, as time passes, you start gaining a lot of marks over the DNA. Let me tell you something. Whenever you gain a lot of marks, this DNA become inaccessible. So your cells cannot read the DNA anymore, and the cells lose functionality. In other words, you get older. So as time passes, you gain a lot of marks, and this DNA is not the cells are not able to read this DNA. As a consequence of that, you get older. Yeah. And remember, there is a trick to avoid this. There are three proteins that are called together OSK that revert this effect. So they clean the DNA from all these marks. So it reverts. And this technology is available now. We are able to revert the DNA, the aging of the DNA. In other words, you go back to your 20s, to the health of your 20s and the appearance of your 20s. Okay, this is the whole purpose of this technology, the DNA rejuvenation technology. Let's go for a little introduction, and after the little introduction we start with today's topic. In this slide is a summary of the whole process necessary to produce EV-OSK with the three DNAs corresponding to the three proteins, the OSK proteins, the proteins that rejuvenate you, okay? And today's topic is cleaning or purifying the EV-OSK, which is the very last step. So congratulations, you got here. Okay, let's go for it. There are three steps in the production of EV-OSK. Okay, once we have the DNA, remember the DNA with OSK, with the OSK genes that are necessary for the OSK proteins. Once we have the RepCap DNA, this RepCap DNA is the DNA necessary to form the virus. So here we have the DNA for the virus, here we have the DNA for the proteins, and here we have another DNA that just help the process. Okay, so the first step is producing many copies of these three DNAs. Okay, so we introduce each DNA in a bacteria cell line, and we grow the bacteria. And as the bacteria grow, it duplicates the DNA. In other words, it produces many copies. So the first step was producing millions of copies of DNA in bacteria cells. Okay? The second step was the DNA OSK is expanded and encapsulated inside the AAV-OSK. So the DNA with the three proteins is expanded and encapsulated in the virus, okay, inside the virus, and the virus will deliver the DNA of the three proteins to the cells, in the human cells. What do we do? After producing millions of copies, we just kill all the bacteria and extract the DNA. And once we have the three kinds of DNA, we mix the three DNAs and include it in another type of cell, mammalian cell. This is a mammalian cell. You see it here? So here we have the three types of DNA that are included in mammalian cells. Okay? The three DNAs will go to the nucleus of the cell, and it will duplicate the OSK genes, and it will produce EV particles, EV virus. And the own cell is very clever, and it will include the genes inside the virus, and it will release the virus with the OSK genes to the media, to the out of the cell. Okay, so here we have the AAV-OSK ready. Ready? No, we need to clean it. So the third step in the production is the purification. So we will purify, we will clean this AAV-OSK, we will clean it, we will purify it, so we will get rid of contaminants, so that is safe for the human beings. Okay, so that is today's topic. Today's topic is cleaning further the AAV-OSK. Okay, are we ready to start? Let's go for it. We are absolutely ready to start. In today's topic, we are cleaning the AAV-OSK by using a technique that is called ion exchange chromatography. And the principle behind this technique is the ionic interactions. You remember from the school, this is easy, okay, that the molecules are charged, okay, so molecules are positively charged and other molecules are negatively charged. Okay, you can see here the molecules. When you have a molecule that is negatively charged and another that is positively charged, they attract each other. Okay, so whenever you have different charges, they attract each other. What happens when they have exactly the same charge? It doesn't matter whether they are negative or positive. You see here, when both charges are negative, yeah, they repel each other. Is the same case when they are both the molecules are positively charged. They repel each other. They don't want to they stay together. Okay, so AAV-OSK, you see here, AAV-OSK is negatively charged. Let's come here. So it's negatively charged, so it will attract positive charge, so it will get bound to positive charge. Okay, so let's see this. This is the point in which we are now. We have AAV-OSK that is negatively charged with some other contaminants. You see here, some of the contaminants are positively charged and other contaminants are negatively charged. So what we are going to use is a resin that is positively charged. We will have our sample, and we will use our sample with a certain resin. It's a column that has resin inside that is positively charged. So as the sample goes this way, negative components of the sample will bind to the resin, and other components of the sample that are positively charged will be excreted. Okay, let's see here. Once you pass your sample through the column, the positive contaminants, we will get rid of the positive contaminants because they will not interact with the positive charge. They will be repelled. And what happened with EVOSK? EVOS will get stuck in the resin with other contaminants that are negatively charged as well. You see here, this is the resin that is positively charged, and you have EVOS attached to it and other negative contaminants attached to it. So just by this step, we have cleaned it a little bit. And well, in some occasions, only by this, you clean a lot. But still, you have some negative contaminants and EVOS. And what is the way to eliminate these little contaminants? Okay, we start increasing the amount of salt in the media. So we start adding little by little, increasing quantities of salt. And as you increase the quantity of salt, some of the contaminants will be released, or maybe us. The point is that by increasing little by little, we will counteract uh, the force by which the contaminants or EVOs is attached to the column. So in other words, we will separate. As we increase the salt, the different components that are here inside the resin will be released. So by increasing a little bit, maybe one contaminant will be eliminated. By increasing a little bit more, maybe another contaminant or maybe EVOs. The point is that we are able to separate the EVOs from some contaminants, but increasing little by little the salt. Yeah, whenever you want to release everything inside the column, you just increase very much the concentration of salt, which is this case. Okay, so by increasing little by little the salt, you release separately the different components of the sample. So this is the main principle of this technique, is just increasing little by little the salt so that you can separate EVOs from other contaminants. Understood? I hope so. It's just it. It's just using this property of EVOs, that is charge, okay, to separate the EVOs from other contaminants. Well, and now that we know what ionic interactions are, we are going to understand what chromatography is. And this is what this slide is all about. Chromatography. We are going to understand chromatography. Let's read with me. Chromatography is a variety of techniques used to separate different components of a sample. That means separating the target protein of the target, which is in this case EVOSK, is separating it from the contaminants. Okay, and the ionic chromatography separate components based on differences in charge of the components. So we are going to separate the EVOs from other components or other contaminants using the different ionic interaction forces. This is kind of a chromatography equipment. This one in particular is called Akta. Okay, this is how it looks like. And for the ionic exchange chromatography, we use 3 steps. A first step that is the loading. So we load the sample in the system, okay? So a pump pushes the sample in the column with a positive resin, and the AAV-OSK binds to the column, and the positive contaminants are discarded, okay? Let's see it a little bit here, okay, step by step. Okay, this step would be the sample loading. You see here the sample. Okay, in the sample we have the EVOs, which is negatively charged, and other contaminants. And here we have the pump. The pump pushes the sample towards the resin. You see here? Okay, so EVOSK will get attached to the resin because the resin is positively charged and EVOSK is negatively charged, so it will get stuck here, whereas the contaminants are expelled. Okay, so EVOs will remain here and the contaminants will go through the column without interacting with the column. And in the chromatogram, which is the representation of the signal, will be like this. It will increase in signal. Why? Because it will record proteins, okay? And there will be a lot of signal because of the contaminants. So the signal will increase due to the contaminants. You see here, it increased. So this is the first step. This is the loading. So we pump the sample with EVOs and contaminants. EVOs will remain in the column, whereas the contaminants will elute. And we will see the contaminants here as an increase in the signal. Okay, what happened next? The second step is the wash. We clean further EVOs bound to the column. Okay, so we use a buffer that is low in salt. We call it the buffer A. Okay, this is a buffer with a low concentration of salt. You see here we have another pump and it will pump the buffer through the column. Okay, so a small quantity of contaminants will elute, just a little quantity of contaminants. As you see here, as we pump the buffer, the signal will decrease because the contaminants have disappeared in the previous step. So they will disappear little by little by little by little by little. And in the column, it only will remain EVOs, which is negatively charged, and this time a tiny small quantity of contaminants that are negatively charged, as it is the case for EVOs. You see here, right? So this is the wash. We wash the wash buffer A, clean EVOs further from contaminants. Okay? And the last step is the elution. What do we do in the elution? We increase gradually the concentration of salt. By doing so, we separate negative contaminants and OSK. Okay, so we have here, as you can see, the buffer A, and here we have the buffer B. There will be two pumps, one pumping buffer A and another one pumping buffer B through the column, and in time the amount of buffer B that is pumped increases, so the concentration of buffer B or salt increases. Okay, so as we increase, we start having different peaks, and the peaks will correspond. One of the peaks will be EVOs here, and another peak will be the contaminants. So by doing so, we separate EVOs from the contaminants. Okay, so let's read with me. Elution gradient. The amount of salt is increased gradually by increasing gradually the amount of buffer B pump. Each component of the sample leaves the resin at a specific salt concentration. They will be separated, and we will see them as separate peaks. As you can see here, we will see the contaminants and the AAV-OSK separated in different peaks. So this is how a chromatography works. This is in particular ionic exchange chromatography. It uses ionic interactions as a principle for this chromatography, and the chromatography is just a variety of techniques used to separate different components in a sample. Okay, so let's go for it. Let's go for the technique. I will explain step by step how we perform a ionic exchange chromatography. Well, very easy. This technique is performed in only 4 steps. Only 4 steps? Yes, only 4 steps means there's no summer. Okay, let's read with me. Okay, let's go for the 4 different steps. The first step is sample preparation. The only thing that we do is diluting the sample in salt, diluting the salt of the sample. Okay, you see here the sample has a big a high concentration of salt. What we want is the sample with a small concentration of salt. Okay, so we prepare a dilution buffer, that is a buffer without salt, and we dilute the sample with the dilution buffer to reduce the amount of salt. That is to say, we just have our sample, we just have the dilution buffer, and we add dilution buffer so that we dilute the sample. So this is the first step. The second step, what it is preparing the buffers, the buffer preparation. Do you remember the chromatography in the previous slide? In the chromatography, we were using 2 buffers, right? Buffer A and buffer B. Buffer A with a small concentration of salt, and buffer B with a high concentration of salt. So the second step is just preparing the 2 buffers. Okay, buffer A, low salt, buffer B, high salt, high concentration of salt. And what about the step number 3? Okay, let's read with me. The number 3 is chromatography system preparation and setup. So we just prepare the system, the Akta system, the chromatography system, and we set up the program. Okay, so we prepare the system, we place the ion exchange column, the sample, and the buffers in the correct place. I will show you. And then we set up the program, which includes an equilibration, that is just preparing the system, a load of the sample, as I told you, a wash of the sample, and dilution of the sample. Okay, so this is the number 3, is preparing the system and setting up the program that will run the chromatography. What is the number 4? Very easy. It's just running the ion exchange chromatography, and the only thing that we are going to do is press start. Press start in the software. So by pressing start, after pressing start, the system what is going to do is equilibrating, and equilibrating means rinsing first in buffer B, afterwards rinsing the system with buffer A through the whole system and the column. Then it will perform the loading, as I have told you. Then it will perform the wash of the sample, and then it will perform the dilution of the sample, which will be done by increasing the concentration of salt liter by liter, and that is done by increasing the proportion of buffer B that runs through the system. Okay, easy peasy. Let's go step by step. Here we are in the step number 1, sample preparation, which is salt dilution, which is diluting the sample in salt. Okay, we will go from a sample with high concentration of salt to a sample with a small, little, low concentration of salt. Okay, So what we are going to do is preparing the dilution buffer, which has no salt. Afterwards, what we are going to do is diluting the sample with the dilution buffer to reduce the amount of salt. In other words, we prepare the buffer with no salt and we add dilution buffer to the sample to reduce the salt. That is all. So, but let's have a look, okay, on how we do it. So we prepare the dilution buffer without salt. This is the dilution buffer, okay? So we dilute the components. We have the components, okay? We dilute the components. We adjust the pH to pH 7.2, which is a pH in which the sample feels comfortable. We filter the buffer with the 0.2, 0.4 microns filter, and after filtering the buffer, we consider that it is clean or is sterile, and we store the buffer at 4 Celsius degrees in the fridge until it is used. Okay, so in here we just prepare the buffer, and I guess you know how we prepare is diluting the components. Here you see the components. If you want to resort to the recipe, it's here, but it's very easy. It's a pH of 7.2, 20 millimolar of sodium phosphate, 1 millimolar of magnesium chloride, and 1% of glycerol. Well, the sodium phosphate will be the component that keeps the pH, okay? The magnesium chloride kind of stabilizes the DNA. So by stabilizing the DNA, it doesn't interact anymore with the AVOs. So we could get rid easily from the DNA, from the remaining DNA, which is a contaminant in this occasion. 1% of glycerol. The glycerol stabilizes some of the forces in the surface of AV-OSK. So by stabilizing the forces in the surface of AV-OSK, it is happy alone. So it helps the cleaning process. Okay, so we prepare the following setup to dilute. Okay, you see here, we place the sample on an ice bath. Okay, so by doing so, we keep fresh the sample. And you see here, this is the sample, right? The sample has the AV-OSK and some contaminants. And we include here a little magnet, okay? And this little magnet, why? Because we have a magnetic stirrer. You see this machine? This machine has a motor that rotates, and it has magnets, strong magnets here. So by rotating the motor, what it does is making the magnetic stirrer to rotate. So at a slow pace, okay, at a slow speed. So we will have here the sample steering, okay, and it looks like this. So here you can see the ice bath, which is just a box with ice, and we place there the sample. You see here, and this is a magnetic stirrer. You see here, this is the speed knob. Okay, so by manipulating this, we can increase or decrease the speed, but for this purpose, we need a slow speed. Okay, so we prepare a nice box, transfer the sample to a beaker placed on the ice box. As you can see here, this is the beaker, this is the ice box, and we place in the beaker a magnetic stirrer. So we just include here a magnetic stirrer, and we place the magnetic stirrer and we just give some speed, so it will start the stirring. Okay, and dilution, yeah, as it is rotating, as it is stirring, we just add little by little the dilution buffer. This buffer that we prepare here, we just add little by little the dilution buffer. We transfer it with a pipette, with a big boy, which is a big pipette, or however you want to do it. Okay, so we collect the dilution buffer, which is here, okay, which is also refrigerated. we collect the dilution buffer and a pipette. You see here, this is my pipette. Remember, the dilution can be done by adding buffer at a pace of 10 to 15 milliliters manually or with a pump. This thing that we are doing with our hands, it could be done by a pump that adds little by little the dilution buffer to the sample. Okay, but it's just treating well the sample. This step involves just treating well the sample. So we just transfer little by little the dilution buffer while we keep it fresh. Okay, so that is all that it is. The first step, very easy, is just diluting the sample with a buffer low in salt. And here we are in the second step, which is super easy. It's just buffer preparation. The preparation of the buffer A with low salt and the buffer B with high salt. Okay, let's have a look on the components. You see here, this is the buffer A, which has a small concentration of salt. So it just have basic components. The pH is the 7.2, okay? And it has sodium phosphate. As I have told you, this is the component that keeps the pH steady. This is the magnesium chloride. This is for stabilizing the DNA. So in other words, being able to get rid easily from DNA. And 1% of glycerol. Remember, the glycerol keeps the protein happy. Okay. And what about the salt? As you can see here, there is no salt. There is a small concentration of these salts, but no sodium chloride in excess or something. And have a look here. The buffer B. We have the buffer B, which in this case has a high concentration of salt. In this case, 350 millimolar of choline chloride. Okay, we could use sodium chloride instead, potassium chloride instead, but for the purpose of separating the components of this sample, it turns out that the choline is very, very good. So let's have a look on the components of this salt, of this buffer. We have a pH 7.2, which is exactly the same. 20 millimolar of sodium phosphate, which is exactly the same. 1 millimolar of magnesium chloride, which is exactly the same as the buffer A. 1% of glycerol, exactly the same, but in this occasion we have a lot of salt, 350 millimolar of choline chloride. Okay, and okay, for those of you that are not familiarized with the buffer preparation process, let's have a look at here. Okay, so the first thing that we do is we have all these components in powder, so we weight the components by using a scale, a balance. Okay, a balance looks like this. So we have put here the components, we calculate the amount of components that we need, and we weight the components. After weighting the components, we just add the components to water. Okay, so we will have a cylinder here with some water. We use normally for starting diluting the things, we use 50 to 70% of the final volume in millicube water. So we will have our cylinder measuring the volume. We will put half the volume with millicube, and we dilute the components in the millicube water. Millicube water is ultra pure water. Okay, so after diluting the components with a magnetic stirrer, we dilute the components very well by shaking. For example, once we have all the components here, we dilute the components as much as we can, and we do it by shaking, by rotating with a magnetic stirrer. Do you remember the magnetic stirrer machine? This is the magnet that we use. We use a small magnet that include inside the cylinder, and this is another machine. Okay, this is another steering machine. that will rotate and will make the magnet to rotate, so and the sample will be stirred. Easy. So once we have all the components well diluted in the sample, what we do is adjusting the pH, and we use, in order to adjust the pH, a pH machine. You see here, this is the probe of the pH meter. So we include the probe of the pH meter. You will see here the value for the pH, and if the pH is greater than 7.2, we add some acid chloride. So by adding acid chloride, we will reduce the pH. What happens if we saw the pH, a low pH, let's say under 7.2, let's say 6.7? We just add a basic base. So in this case, normally we use sodium hydroxide. Okay, so the sodium hydroxide will increase the pH. So once we have a pH of 7.2 or close to pH 7.2, what we do is filtering the sample. So we have here after adjusting the pH, we adjust the volume. So we will top up the buffers up to the final volume, and we use water. Okay, so once we have our buffers well prepared, we want them sterile. So in order to filter the buffer and get it sterile, we use filters. Okay, so we use a 0.2 micrometer filter. So the pore of the filter will be 0.2. With these filters, we guarantee that there is no bacteria or something in the buffer, that the buffer is absolutely clean. So we filter it with this device. You see here a funnel, so we include the sample in the funnel here, and this device has here a little a little connection with a pump. So, and this is the pump. Okay, so we activate the pump, it will make a vacuum, and by doing vacuum, it will pushes the sample through the filter, and it will pour here at the bottom of this bottle that it is sterile. So once all the sample is in the bottle, sterile and clean, we storage the buffer in a fridge at 4 °C, and it will be ready to work with. Okay, so this is this step, is just preparing the buffer A and the buffer B, and you have here the receipt. You will see also the receipt in the website. Okay, so take it easy peasy. I have extended very much in this slide, but in fact it's very easy. It's just preparing the buffers. So let's go for the step number 3. Okay, here we are in the step number 3. The step number 3, remember, is the chromatography system preparation and the setup. We are going to prepare the system, and we are going to set up the program. Okay, this is the number 3, chromatography system preparation and setup. The chromatography system is called the Akta Pure. This is just a brand, okay, and it looks like this. This is the device. It's just a system with different, seems it is very fancy, but in fact it's just pipes and pumps, okay, and we attach things like the column with the resin. So we are going to prepare the system, which is just placing in the correct place the ion exchange column. You see here the ion exchange column, and the ion exchange column looks like this. We are going to place the sample and the buffers in the correct place. That is all that is involved in preparing the system. It's just placing the column in the correct place, the sample and the buffers. Okay, so let's have a look. Let's have a look here. Okay, you see here, this is the sample. Okay, this is the sample, and what we do is placing the sample line Okay, so in this system there will be a pipe that is called sample line. We will see it because it clearly states, it says sample line. You will identify it because it will say sample line. So the only thing that we have to do is identifying the line, which is the sample line. We will place the sample line, the sample pipe, inside the sample pump. Okay. So that is everything related to the sample preparation. It's just leaving the sample with the sample line. Okay, you see here the sample line has a pump that will pump the sample through the column. Okay, and what about regarding the buffers? Okay, there will be another two lines. It will be the A line for the buffer A and the B line for the buffer B. Remember, the buffer B is exactly the same as buffer A, but with a lot of salt. So you see here, this is the buffer A that this is being prepared. So we just identify the lane A. So it will say very clearly here. You cannot see, but it says just the lane A. So we place the lane A inside the buffer A. And regarding the buffer B, exactly the same. We identify the system in here. We look for the lane B. So once we see the lane B, we just put the lane B inside the buffer B. Easy peasy, right? Until now is very easy. It's just identifying the sample line, put it inside the sample, identifying the lane A, put it inside the buffer A, identifying the B line, and put it inside of the buffer B. Okay, the last thing that we have to prepare is the column. You see here, this is the column with the resin, with the negatively charged resin. Okay, so we just have to identify this. You see here, this is kind of a valve, and this is the line for the column. So the only thing that we have to do is using this line to screw, to attach it to the column. You see here in this occasion, since we are using a lot of sample, we need 3 columns. In this case we are using 3 columns in row, but majority of the time you will be using just 1 column. The columns look like this. It's exactly like this, exactly what you see here in the picture. So up to this moment, what we have done is just placing the sample where it should be, placing the buffer A with the lane A, placing the lane B inside the buffer B, and attaching the column. Okay, so that is all. That is all. The sample will be pumped first, then we will clean with buffer A. We clean from contaminants, and then we will increase the buffer B. So the pump will start increasing the concentration of buffer B, and the EVOs will be eluted. Okay, this is how the magic works. And as what comes out of the column will be collected, okay, in these collector plates. It's a plate with a lot of wells, as you can see here. These are the plates. In this occasion we are using 5 plates. Okay, this is the collector. Okay, so everything that comes out of the column will be collected in different wells. Okay, this is the fraction collector. It's called fraction collector, and it will have inside the collector plates. You can see this unit here easily here. Okay, so let's have a look. Normally we put the buffers and the sample here, as you can see. Then the columns are attached here in this area, and then we have the collector that is normally is a side. You see here the fraction collector, easy peasy, right? Okay, we have kind of a zoom in the system like this. As you can see here, we have we have put in this occasion the sample here. Well, no, sorry. The sample is here, okay? and then we have the buffer A and the buffer B, and then we have here the collector. This is another setup. So this is the correct way to prepare the system. It's just placing the sample, placing the buffer A, placing the buffer B, placing the column, using new collector plates. And what we are going to do now is setting up the program. Okay, for setting up the program is very easy. It's just kind of a recipe. We just have to follow a protocol that is very easy. It's just reading and setting up the program of the system. The program is called Unicorn. You will be able to resort to these protocols by going to the website. Okay, it's because it's just a recipe. It's very easy. You just have to follow the steps. In 5 minutes is done. But it's a little bit long, so I rather include it into the website. So whenever the website is ready, you will be able to access the program, how to set up the program. But the program is just a set of orders that will include the loading, the wash, and the elution. That is all that you need to know. Okay, it's an easy program that runs this process, okay, of loading, washing, and eluting. So let's go for the fourth and last step. Well, here we are almost at the end of today's session. Okay, the ion exchange chromatography. This step is the easiest one because yeah, I will tell you, this is just running the ion exchange chromatography. So let's read here with me. Step number 4, running the ion exchange chromatography. And what do we have to do is just pressing the start button and the program will run. So that is the only thing that we are going to do, is going to the software and pressing start. And the program will run the different steps. Okay, the different steps you know them because you saw it in the introduction slides. But anyway, let's go for it. It's kind of a repetition. Okay, so once you press start, which is the only thing that you have to do in this step, the program will do as follows. It will load the sample. So loading the sample into the column. You remember you place the sample in the sample lane, and the program will say to the Akta system, pump the sample. So it will pump the sample through the column. The AAV-OSK and another negatively charged contaminants will get attached to the column, whereas the contaminants will be released. You see here, the signal will increase due to the contaminants. Okay, and once the system detects that the sample has been fully loaded into the column, it will go to the following step. And how does the system detects that you have loaded everything? It detects that you have loaded everything because there only will be bubbles in that is air in the system. So the system will detect the air. So that means that the loading has finished. So once the system detects that the loading has finished, it will start the following step. The following step is the wash. So we use buffer to clean a little bit. Okay, the program will use buffer A without salt. It will pump the buffer A through the pump through the column. So it will wash a little bit the EVOs that is inside the column. At this stage, there only will be a tiny small quantity of contaminants, negatively charged contaminants. Okay, so it will look like this. You see here. Now it's just pumping the A. You see here. This is an actual picture of the software. So it will just pump the A buffer. Okay, so as we pump the A buffer, you will see as the signal decrease. That means that the amount of contaminants has been reduced very much, so we will have no signal. Okay, the signal will be close to zero. You see here, the first step, there was a lot of contaminants, well, a little bit of contaminants, then they decreased to. So at the end of the wash, what you have inside of the column will be just the AV-OSK and super small amount of contaminants. So how does the program know that it has finished? Because we set in the program a certain amount of buffer A to be pushed. So once this step finish, we go for the last stage, which is the elution. We use buffer B to increase the salt gradually and separate EVOs from the last of the contaminants. Okay, so here we are. This step was finished, and now at this stage, we will set up in the program a gradient of salt. That means that over time the amount of buffer B will increase, and by increasing the buffer B we will increase the amount of salt little by little, gradually. So as we increase, you see here the amount of salt of the buffer B. This blue line represents a gradient, a salt gradient. Okay, the increase in salt concentration. So as we increase the salt, as you see here, the different components of the sample will be eluted. So sometimes the AV-OSK elutes early, or sometimes the contaminants. In this occasion, you see the contaminants which are here, you see represented here, elutes first, and afterwards, after increasing a little bit more the salt, the EV-OSK elutes, and both of them elute separately. So at the end of this process, we can consider that EV-OSK is 99 or greater than 99% pure. It's super pure, super safe to be used by humans, or in whichever experiment you want to use the AV-OSK or another kind of proteins. Okay, so that is it. At the end of the process, you will have the EV-OSK completely clean from contaminants, completely clean. Okay, so that is what happens after you press start. The only thing that you have to do in this step is just pressing start and wait for the magic to happen. And the magic is just this. I have included real pictures of the software. Okay, this is what you will see. You will see, for example, in the loading, you see here, this represents the sample line, and it goes through the column, the pumps, and it goes here to the collector. Okay, this is what happened in the loading. Then, as you can see here, you see this represents the A line. So you will see the A line in green. It goes through the column, and it ends up in the fraction collector. And in the last step, you see that the A line and the B line are highlighted in green, meaning that both of them are working, but at different percentages. They go through the column, and it ends up in the fraction collector. Okay, these are real pictures of what you will be seeing on using this technique on an Akta system. So that it is. That's all, folks. The 4 steps explained. You can resort to the website. It will be, these slides will be in the website, along with the protocols, with the real protocols, describing step by step. In this in the website, you can see every single detail will describe. Okay, so let's go for the summary. Here we are in the summary. Yeah, the session has finished. At this point, you have very clean and nice EV-OSK. In this occasion, yeah, really clean, completely purified, completely safe to be used. Okay, very nice EV-OSK, clean. Okay, So you have the virus with the DNA inside, the DNA that has the OS genes ready to rejuvenate your DNA, ready to make you go back to your 20s, to the young of your 20s. So let's remember the 4 steps. Remember, the first step was sample preparation. That was just diluting the salt by adding dilution buffer. You see here, right? So it was just diluting the sample to reduce the amount of salt by adding dilution buffer with no salt. Second step, preparing a buffer A with no salt and preparing the buffer B with high salt. Step number three, chromatography system preparation and set it up, which was just placing the sample line inside the sample, the A line inside the buffer A, the B line inside the buffer B, just placing the columns, the column with the negatively charged resin in the correct place and setting up the program. On the program, you can see how to set up the program in the website. Very easy. It will take you no more than 5 minutes to set up the program. Okay, so it's so easy that I have just described it in the website. It's just reading a protocol, a one-page protocol. And the fourth step, which is just pressing start, my friend. You just have to press start and the program, the system will do everything for you. It will load the sample, it will run buffer A to clean, it will run that was the wash, and it will run afterwards the elution stage, which is increasing the buffer B, increasing the salt, and the EVOs will elute separately, very clean from contaminants. That's all, my friends. Okay, so this protocol will be well described in detail in working instruction or SOP, standard operation procedure, with the following name: 34 WI006 ion exchange chromatography on Akta systems. Okay, and these protocols, these working instructions or SOPs, are compliant to CGMP for quality, ISO 9001 for quality, ISO 45001 for health and safety, and ISO 14001 for respect of the environment. Okay, so these protocols are fully compliant with these quality systems. Okay, so you can fully you can fully use them, you can fully implement them in your company. Okay, so they are ready to be used in industry. Okay, and you will find them in the www.novosumer.com, the website that will be available in the following months. Okay, you will find everything well detailed and very easy, ready to use. Okay, so all this technology will be described there for free access, free everything. Okay, just for you to enjoy. Thank you very much, my friend. Yeah, Novosumer says thank you very much for watching it. Thank you very much. I will be uploading other protocols, but basically all the technology has been described in these 7, 8, 9 videos. The rest of the videos that I'll be uploading is just quality control checks and extending a little bit the information that has been offered in these 7 to 9 videos. I don't even remember the number of videos. Oh my gosh. So let's enjoy, my friend. Let's enjoy. This technology is there already available. You will see it more and more in the news, in the economy index and these sort of things, because this is the present and the future is already here. So enjoy it. Okay, I hope you are an entrepreneurial spirit and you implement these protocols and you get rich and you help the rest of the humanity. Be good, my friend. Enjoy the life. Okay, see you soon. Bye bye.